All right, I think we can go ahead and start. Welcome to this week's Connect Science seminar. I'm very excited to introduce our speaker today, Dr. Owen Whitty, a university professor at UCLA, President's Chair, Developmental Immunology. He's also the Director Emeritus of Ellie and Edith Broad Center of Regenerative Medicine and Stem Cell Research, and the co-director of Parker Institute of Cancer Immunotherapy at UCLA. Dr. Whitty is an elected member of the National Academy of Sciences and a former HHMI investigator. Dr. Whitty is really a distinguished leader on identifying key drivers of cancers, and some of his most significant contributions include the discovery of the tyrosine kinase activity of the ABL gene and demonstrating the role of BCR-ABL in leukemia, which ultimately resulted in the development of Gleevec. He also co-discovered the Burton's tyrosine kinase critical in the B lymphocyte development. And more recently, his group has been focusing on defining the stem cells and growth regulatory pathways for epithelial cancers of the prostate and perhaps also other organs to help define new types of therapy for these diseases, with particular emphasis on immunotherapy, which is what we'll be hearing more about today. Before passing it off to Dr. Whitty, I'd just like to point out one housekeeping thing. Please type your questions not into the chat option, but into the Q&A option, and we can have Dr. Whitty answer them towards the end of the presentation. This meeting will be recorded and will be posted on our website. With that, Dr. Whitty, thank you for being here today. Please take it forward.
All right, thanks for the introduction. Always nice to visit at Dana Farber, even if it's by Zoom. And I'll look forward to particularly meeting with the postdocs and students later this afternoon in more of a discussion format. You can actually see who I'm speaking to. So let me start by, why is my slide not advancing? There we go. All right. Let me start by saying that I do a lot of consulting in the biotech world, and many of these companies are very interested in the area of immunotherapy of cancer. But all the work I'll talk about today is supported by normal academic means, philanthropy, and of course, money from government sources as well. So since I know people get distracted during Zoom talks, let me give you a summary of the presentation, and then we'll come back at the end to discuss each of these points. First, we're going to talk a little bit about how we model the progression of aggressive prostate cancer using a human epithelial cell transformation model. In collaboration, my former fellow, Jim Park, now at Duke, and my good colleague, Tom Graber here at UCLA. I'm going to talk to you about how we've been developing T-cell receptors to target prostatic acid phosphatase, hopefully eventually to bring these into the clinic to treat patients with earlier stages of prostate cancer. This is in collaboration with a number of labs at Fred Hutch, Chris Garcia at Stanford, James Wolfschlegel here at UCLA for Mass Spec, and Jim Heath's lab also in Seattle. And next, defining late-stage prostate cancer-specific splicing products, which are presented by different HLA alleles. This is an attempt to really expand the range of potential targets that we might use in treating advanced prostate cancer and other related forms of cancer. And then finally, I'll tell you about some recent work just recently submitted for publication, in which we've looked at a new technology developed by the Dino DiCarlo's lab here at UCLA, which is hydrogel-based nanovials in which we can isolate specific T cells with, I think, very high sensitivity and specificity. But to start, I have to tell you that this work and idea of finding targets for cancer, for me, goes back about 50 years to when I graduated from college at Cornell. At that time, President Richard Nixon was not a beloved figure by any means, and I could hardly imagine I would ever have anything nice to say about him. But he did one very important thing, which is that he signed the National Cancer Act. And among the benefits of this was major new funding to study oncogenic viruses of various species, including mice, rats, chickens, etc. This really stimulated the field of molecular oncology, defining all kinds of genes and activities that cause cancer and could later turn into potential targets. When I went off to medical school, I worked with Irv Weissman at Stanford, still going strong, running their stem cell center as well. I studied this particular virus and for several good reasons. This is the Maloney-Murine sarcoma leukemia virus complex. We now know it's made up of a mixture of viruses, one replication competent, shown here as MMULV, another replication defective, but carrying a unique oncogene called MOS. This was a time of great discovery of this families of oncogenes, and they all had very useful activities, which then connected to cellular genes that we now use as targets. But what was most impressive about this system was the very dramatic way in which inoculation of this virus into the muscle of susceptible strains of mice or rats could rapidly cause the growth of a very large tumor. But equally as impressive was that in specific strains of mice or rats, there would be a robust immune response which would eliminate this tumor and protect the animal from subsequent re-challenge with the virus. We thought that if we could understand the pathogenesis, what drove the production of the tumor, and understand what caused its regression, we might have an analog or an idea of how to deal with human cancers, perhaps driven by similar genes. This idea turned out to be true, but it took an awful long time to understand the specific set of genes like MOS and for the knowledge of the immune system to be enhanced so we could understand what cells and what mechanisms participated in this dramatic regression. No, decades later, that the specific regression in this case is actually caused by a very strong epitope coming from the GAG or antigen gene of the virus, which when presented in the appropriate mouse H2 gene can engender a very, very strong immune response and rejection of the tumor. So we'd like to be able to do this in different types of human cancer. It was alluded to in the introduction, but this idea of searching for mechanisms that drive pathogenesis can be very successful if that particular mechanism is connected to a particular biology. And the two very good examples that came from work in my lab, BCR-ABL gene and its eventual development of the drug imatinib and now 4 or 5 follow-on compounds, as well as the drug Bruton's tyrosine kinase and a family of drugs exemplified by ibrutinib. In both of these cases, the reason these are good targets and effective therapies is that the cell is dependent on these particular proteins and mechanisms for its continuous growth. And you have to find a situation in which whatever the target is, the cell is either dependent on it or will continue to express it if you're going to attack it with the immune system. And so our goal, as we transition through the about 20 years of work in my lab, was to continue to find new mechanisms that we could attack, and particularly, I'll tell you about today, attack with immune-based mechanisms. About 25 years ago now, I got interested in prostate cancer for a number of reasons, including members of my own family were diagnosed with it, and began to study this as a new area of investigation with my colleagues, Charles Sawyers, who was then at UCLA, and Rob Reiter, another trainee in my group who's still at UCLA as a senior surgeon in urology. There was a paucity of models to study this disease at that time, and one of our first goals was to prepare new xenograft models, the LA series, which is still in use today. And a second was to begin to look for uniquely expressed proteins that we might use as therapeutic targets. One such protein, prostate stem cell antigen, or PSCA, is still being investigated as a target in a number of companies and a number of academic centers as a potential treatment for advanced stage prostate cancer. What we really needed to do, I felt, in order to understand this disease was to develop model systems that we could genetically manipulate using human epithelial cells as targets for the transformation process. A very important step in this process came from the work of Andrew Goldstein, former student who's now on the faculty at UCLA, in which we combined elements of systems we had worked out in using murine tissues, but in this case using human epithelial cells from the prostate, to transform them with a combination of activated genes such as AKT and ERG. particular combination wasn't so critical here as the fact that we could drive these human basal epithelial cells into a model of prostate cancer, which could grow in mice, immune deficient mouse models, that we could then study combinations of genes and think about the biology of the cancer. The problem with this particular stage of the model was that it grew very slowly and didn't represent the more aggressive, more active model of cancer we were most interested in trying to develop new therapies for. And so the overall feeling about prostate cancer has to take into account this very stereotypical pattern of disease in which local disease can be treated very effectively with either prostatectomy or radiation therapy, but that over some period of time, many patients will develop metastatic lesions, which can be treated with various forms of castration therapy, either blocking the production of hormone or blocking the production of hormone action. This is amazingly effective in most patients, and the adenocarcinomas will disappear. PSA, the marker we use to follow this, will go down to very low levels. And then for some relatively unclear period of time, where you can't really predict who's going to regret or return, there will be a recurrence of the disease, which is now castration resistant, still maintaining an adenocarcinoma phenotype for the most cases. But a fair small number of patients will start with the disease that gets the name neuroendocrine or small cell carcinoma features, but that usually is not seen in this phase of the disease that's recurred after castration therapy. But coincident with this very stereotypical pattern of disease recurrence and response to therapy is a remarkably diverse set of genetic events here displayed in a nice data set from Rul Chennai and Charles Sawyers and colleagues back a few years. And I know this is a very difficult slide to see, but the important point is that if you look at any of the genes that are modified here on the left-hand side, like AR, P53, or P10, it's in various combinations across any of the individual patients that are shown across the top. There's really no particular combination that appears over and over again in all of the patients. So great genetic diversity, but stereotypical clinical pattern of occurrence and response to therapy. So how does this might work? Well, we were thinking about this and trying to figure out what to do about it. And as part of a large cooperative study with a variety of colleagues at multiple universities on the West Coast of the United States, called the Stand Up to Cancer PCF West Coast Dream Team, we decided we wanted to collect biopsies of patients who had recurred and were progressing in their cancer despite aggressive anti-androgen therapy. And the very first piece of data that came from these biopsies and analyzed by Jyoti Wang and George Thomas was that the adenocarcinoma phenotype shown here on the left was changing in many of these patients to something they called an intermediate or atypical carcinoma, and then eventually could find regions in these biopsies, what we now call small cell or neuroendocrine phenotype carcinoma. What I saw as a biologist was that this was a continuum or a transition that was going on, macro or reorganization of the shapes of the cells and their organization to one another. And this pattern or changing pattern was seen in multiple patients, despite that dramatic variety in different genetic events that had occurred within them. So what we now sort of observe is that this change after castration type therapy and recurrence of the disease to the castration resistant form can have a fairly large percent of patients' biopsies show these small cell features with neuroendocrine markers, et cetera. So what's going on? What's causing this? This range of 20 to 30% might actually be an underestimate, if you think about it, because when we take a biopsy, we're only getting a small fraction of the tissue that the cancer patient is bearing. And even within those individual biopsies, one could see regions that would be more adenocarcinoma and others with this atypical neuroendocrine carcinoma features. So to study this and think about the problem, taking this slide from a very nice review by Michel Beltran, began to wonder, are these adenocarcinoma cells converting somehow by differentiation process into these neuroendocrine-like cells and maybe acquiring these small cell features, or all these different forms coming from some cancer like stem cell? Well, I don't think we know the complete answer to this, but this set of questions has attracted quite a lot of interest in the field. And there's now paper after paper appearing that implicate one or another gene, transcription factor, set of combinations of genes in this process. Genes like SOX2 in the context of p53 loss of function, RB loss of function, other genes like EZH2, other papers showing very interesting response to control by FOXA1, even particular mutants of FOXA1 that have been described in a subset of prostate cancer patients with castration-resistant disease. And more recently, a targetable pathway of JAK-STAT that may be necessary for converting or altering cells from one stage to the other. What we wanted to do was to take advantage of our strategy of directly transforming human epithelial cells of the prostate into transformed models to evaluate whether we could create such a small cell neuroendocrine-like cancer. So Junmook Park led the efforts in this, and a very successful model system that we've exploited over the last few years. What he did in this case was to take isolated cells from the benign areas from human prostatectomy specimens, isolate basal cells as our target population that have higher viability and can reconstitute into these engraftment structures, and then use a series of lentiviruses to introduce 5 different genetic perturbations. First, enhance the level of expression of the MYC gene, add a copy of the BCL2 gene to resist apoptosis by the cells as they undergo oncogenic stress, an activated form of AKT here with a tethered myrstillation signal that takes it to the membrane for chronic signaling, and a shRNA to knock down the level of RB1 and a dominant negative form of p53. There's a lot of genetic damage here, and we didn't come to these things randomly. It came from a series of experiments. But when you introduce all of these genes simultaneously into these basal cells, do a short-term culture in vitro in an organoid culture model to allow establishment of the genes and their expression, and then place them in xenografts, you get a very interesting result. And here's an example where all of those genes were entered into the cells. And what drives out is very aggressive small cell prostate cancer with neuroendocrine features such as NCAM, chromogranin, and synaptophysin. If you leave out elements of this transformation model, for example, leaving out BCL2, you don't change the phenotype, but you do decrease the efficiency of how fast you get these tumors. But if you leave out any of the other genes, such as leaving out RB, leaving out p53, leaving out both p53, RB, et cetera, you can still get transformation. You can still get tumors growing, but they lose the neuroendocrine characteristics and retain characteristics of a prostate adenocarcinoma. So we felt we had developed here was a very good experimental system to evaluate a particular type of genetic damage, and also eventually to develop and ask questions about what happens in this transition from the more adenocarcinoma form to the more small cell prostate cancer form. And one technique that was very valuable in describing the extent of this set of differences was to evaluate how the chromatin changes that accumulated in the cells were marked. And the take-home lesson is that the loss of function of p53 and RB were needed to dramatically enhance the number of ATAC accessible sites, suggesting very much a wholesale change in the genome during this process. Well, we wanted to study this change in a more particular way, and this is the work of Olga Chen, graduate student in the lab, whose manuscript is now submitted on what we call the Part B time course. And this is a very same experiment I showed you on the last slide, taking prostate cells and introducing the 5 factors that we call Part B, putting them through the organoid culture and then into xenografts, and then sampling them at different times along the tumor growth curve to evaluate the molecular changes in transcription and chromatin structure that have occurred. Here's a very nice series of micrographs just to show that you drive through the more adenocarcinoma form into the small cell neuroendocrine form and some related markers. And the appropriate and important result here, looking at the time course and progression and putting it onto these different molecular progression examples based on the RNA expression data of these tumors, is that we can drive from early stages all the way through to the end and very much resemble small cell aggressive cancers that are found in lung as well as the prostate, suggesting that this sort of epithelial change to the small cell phenotype is not restricted to a single epithelial type, but probably something to consider for many types of cancers. We've demonstrated similar work with bladder epithelium as well. Further studies of this time course enable the description of the different transcription factors that occur through this process of going from the basal to the organoid stage to early, transitional, and endpoint tumors. A number of familiar transcription factors turn up, such as ASCL1, NeuroD1, ALK2, SOX2, and OneCut, that have been described as participating in these types of tumor growth before. So it's a complicated picture, many possible control points along the way. Can further evaluate this system using single cell analysis. And I don't want to make this too prolonged, but just to say that the details show that as you go from early to late, you acquire from epithelial cells through stem-like phenotypes and finally into phenotypes that are more reminiscent of neuronal cells as this transformation model progresses. And you can also see, and I think very important outcome of Alke's work, is that the progression from the early through the later stages of these tumors can actually be represented as a bifurcation into two different classes, class 1 and class 2. And class 2 is represented here in red, particularly by the expression of this ASCL1 gene. So there may be different subforms and different subtypes of this neuroendocrine small cell that we have to think about in terms of alternative therapeutic attack. So with that level of complexity and all of this data to generate and look at targets, we wanted to begin to apply the principles of immunotherapy to this problem. This is not an easy job. There's been wonderful examples of using cell-based immunotherapy, particularly using structures like CAR T cells and attacking extracellular proteins like CD19 and BCMA in certain hematological malignancies. This is wonderful and really been a boon to patients in treatment in this arena. But there's also the use of T cell receptor therapy. In this case, clinically useful targets such as the peptide antigens NY-ESO, MART1, WNT1 are also in current clinical trials. Here you have to worry about HLA restriction. You need a specific peptide bound to MHC in order to have a targetable agent that might give you good clinical outcome. And so we set out to study particularly the use of T cell receptors in this regard, trying to find new targets and new TCRs that we might credential and eventually take into therapy. In the particular instance of prostate cancer, we can kind of divide our efforts down into looking at public antigens, such as a prostate tissue antigen, when the cells and the phenotype of the tumors are more like the original prostate. As the tumor progresses to more and more aggressive phenotypes, we may need to use new antigens that are, as I'll describe, that may not be restricted only to prostate cancer, but occur during this conversion to castrate-resistant form. And then finally, as the cells acquire this very aggressive neuroendocrine-like phenotype, we may need to think about these restricted tissue antigens or semi-private antigens, as well as neoantigens, which I won't really have time to talk about today. How do we go about this? How did we get started? Well, the first thing, antigen we took on was prostatic acid phosphatase, which we think is a very good target for immunotherapy in prostate cancer. The expression of PAP is very, very specific. It persists all the way through advanced metastatic castration-resistant disease in most patients. There's specific isoforms of the disease. And most importantly, this antigen was a target for the immunization with a portion of this protein in the drug Provenge, the first cancer vaccine that's been approved for use in patients, where it doesn't have a huge effect in terms of therapeutic effect, but it does extend life sufficiently that's been allowed by the FDA to be sold as a cancer vaccine treatment. But what intrigued us about this is that there was data based on T cell proliferation assays and others assays like ELISpot assays that patients who were injected with Provenge therapy could respond by producing specific T cells reactive to that antigen. It suggested to us that if we could find T cell receptors reactive to PAP, they might be credentialed and be able to use in transfer cell therapy for the treatment of stages of prostate cancer where the antigen is still present. Now, that's a simple idea, but not trivial to make work. And the way we began this analysis was to do a detailed analysis of the immunopeptidome of PAP led by my student Mao, who used 3 different techniques: mild acid elution, a co-immunoprecipitation technique, or a technique that we now favor as the best way to find peptide antigens is a secreted MHC technology. He found 27 different peptides, used those 27 different peptides to immunize or to try to enhance and activate populations of cells taken from the peripheral blood mononuclear cells of normal individuals. And we used a technique called CLINT-seq, which is an adaptation of activation markers which are found in cells which can be fixed and permeabilized. And then after activation in combination with antigen-presenting cells in these different peptides. What makes this CLINT-seq technique works and useful for the isolation of TCRs is that after activation of the cells, they can be fixed and stained for production of proteins like interferon, TNF, et cetera. And then the cells can still be subjected to 10x genomic sequencing because we used a disulfide reducible crosslinker to fix the cells, which when they enter into the 10x genomic sequencing, the crosslinker will break apart and the reverse transcriptase and further DNA sequencing can proceed. From this, we're able to isolate a number of candidate TCRs. They're responsive to individual peptides and interferon by interferon stimulation, and we could test them and credential them in further ways. Frankly, we were very disappointed, however, when we found that the best among the TCRs was just moderately cytostatic for cells, here measured by growth assays of tumor cells and their inhibition with the addition of TCR, peripheral blood mononuclear cells bearing the anti-PAP activity. It basically just showed cytostatic behavior, limited cytotoxicity, possibly because these TCRs were isolated from peripheral blood of normal humans. They wouldn't be expected to be particularly high affinity or high activity because they would likely those cells would have likely been deleted in the thymus by negative selection. So what to do with such a TCR? Fortunately, there are techniques available, one of which have been developed by Chris Garcia's lab called catch bonds. And this is a really simple but elegant technique in which you try to substitute amino acids into the structure of the TCR, which will enable the TCR MHC peptide interaction to undergo an allosteric change in which the new amino acid will actually be captured or caught like a fish hook within the MHC peptide structure and dramatically resist or retard the back reaction of dissociation. You combine this with the avidity and reaction of having multiple receptors on the cell, and it prolongs dramatically the time that TCR and T cells engage with the target. This was remarkably successful, as we can now convert multiple of these ECRs of great specificity in terms of interaction with specific PAP peptides to much more potent cytotoxic activity with this catch bond technique. And we're continuing our investigation of these, testing them in vivo, but our hopes is that some of these TCRs will be credentialed sufficiently to move into preclinical evaluation and eventually into the clinic. So that's one target, but it's clearly not going to be sufficient as tumors evolve and change and lose their expression of PAP. We'll need to have other targets that might appear on later stages of the disease. And to do this, we took on, I think, is a fairly adventuresome project and one that's, again, easy to draw on paper and hard to get to work in practice. There's not that many good TCR therapeutic targets that have been described. This slide was taken 3 years ago. There's certainly some new ones that have been added, like mutant forms of RAS and so on. But again, a relatively small universe of good peptide targets, considering the large number of proteins in the genome. And so what we wanted to do was to ask whether modification of protein structure through alternative splicing and eventually other RNA-based mechanisms might allow for the definition of new antigens that would have high enough specificity that they could serve as immune targets. And to take this on, we needed to work with a number of key collaborators, and particularly Yi Xing, who's a computational biologist. He used to be here at UCLA, and unfortunately for us, he left UCLA and went to the University of Pennsylvania Children's Hospital, where we still collaborate on this project. And we needed his expertise for finding these spliced variants among large data sets. And then 2 other colleagues here at UCLA, Gay Crooks and Chris Seat, who have developed very interesting technologies to grow analogs of thymic-produced T cells in vitro in a technique they called ATO, artificial thymic organoid. And our hope there is that those cells would not be undergoing negative selection and might have a more robust repertoire of cells. Don't know if that's true yet. We're still working on that aspect, but we've made some significant progress in this process of looking for new antigens. If you step way back from all of the things that can happen to a messenger RNA, and there are myriad changes, you can have all sorts of things such as fusions and translocations. You can make circular RNAs, there's RNA editing, you can have transposable elements create new RNAs, but by far the largest number of changes to the genome comes from the process of alternative splicing, which can be regulated by a large number of signal transduction changes. And our hope was that there would be a large number of changes generated from the dramatic signal transduction changes that occur in cancer. To test that, we set out on this project through the collaboration of these multiple labs to have a section on target discovery, which would be the Xing lab and my lab, then to use alternative T cell sources to search for reactive T cells with these predicted peptide antigens we would present to them. This would include peripheral blood mononuclear cells, where most of the data I'll show you today came from, and eventually T cells derived from these artificial thymic organoids. We would use different methods of TCR isolation, including surface activation markers, the Clint-seq technique that I mentioned earlier, and a new technique I'll tell you about at the end of the talk, which are multiplex nanovials done in collaboration with the Dino De Carlos lab here at UCLA. And then once we have those TCRs, hopefully functionally validate some of them as being useful in terms of treating particular types of cancers with expression of those peptides. So finding splice variation is not hard, but finding splice variation at the level that would give you specificity and expression sufficient to serve as a cancer target, that's much harder. And Yi Xing's lab is really a world-leading expert in this. They've developed a number of these algorithms, papers which are listed here, and have used this to interrogate very large data sets from the national data sets of cancer cell lines, specific materials that we've provided them, initially using short read sequencing, because that's the most common data, but more recently including very long read sequencing, which enables you to get specific spliced forms of messenger RNA in a precise order and validate them in a slightly different way. And so the idea of this is a pipeline to take the raw sequence data, modify it with these algorithms, quantify the amounts of RNA of different spliced forms, and then come back and evaluate these different forms and compare them to normal tissues to eliminate those that are expressed in too many bad places you don't want to be treating patients with immunotherapy and causing side effects. And then eventually creating these tumor junction peptides, thinking about how they do or do not bind to specific HLA epitopes, which is still an ongoing concern in the field to define those algorithms to be more faithful in terms of their predicted binding. And then finally, take a look as well while we're looking at the data if something should turn up that might be a CAR T target as well. But I'll concentrate today on what's happening with the TCR peptides. The IRIS program can identify these peptides and you can display them with two important characteristics. What's their tumor specificity, okay? And what's their level of expression in terms of presentation on different classes of tumors. And here's two data sets for castration-resistant prostate cancer or neuroendocrine prostate cancer. And the obvious thing you want are things that are high tumor specificity, but high level of expression if they're going to serve as a good target. And we've been through several iterations of this. There's obviously more and more data being added in the literature to these large databases, more classes of tumors, different papers being published. And so this is an iterative process in which we go, Ease Lab can go over again and again large data sets, hopefully discovering new targets as more data is available for analysis. Each of those particular peptides has to be thought about and compared to what happens in normal cells. There you can use data from the GTEx database and other databases to display the peptides and ask, are they more unique to cancer, or are they also found in different specific tissue sites we'd be concerned about in terms of therapy? So after a few years of running this system, there's some summary numbers. We found about 300 peptides based on their expression and also their predicted binding to HLA A2.01 as our first major HLA type to look at. About 400 TCRs were found that responded in activation assays with the varying selection techniques that I described. We test them in a GERCAT, NFAT, GFP sort of response system as a first approximation. And then that filters down to right now about 17 different TCRs targeting 8 different peptides that we've tested in PBMCs for various activities, including interferon release. Here's one shown here, which is number 238 for a specific epithelial cell splice product antigen 1. And then it's very good. It's giving, 25,000 interferon picograms per milliliter. And here's another TCR called F5, which reacts against a specific MART1 peptide. So it's in the same league as this in terms of interferon response to the peptide. And the F5 has been used in clinical trials with on-site effect in melanoma patients. So we think the process will work to give us TCRs, but it's highly laborious. Many of the candidates drop out, and you have a lot of work to do to credential these. We've taken a couple of them a little bit further. And for example, this one, number 238, redacts to specific peptides which can be processed and presented on HLA-A2 from full-length molecule of this target antigen. And here's showing very nice cytotoxicity over a several-day period with this TCR in ARM PBMCs reacting to a cell line constructed as the test target. There's another one, just to show another example, antigen number 2. In this case, the interferon release is again showing. So each one of these things will require very significant development time to credential, test, and think carefully about in various preclinical models which are underway. So this is about where we are in this target discovery through T-cell sources, TCR isolation, and functional validation. Where we're a little bit stuck right now, and I have to say this is going to slow us down, is that we have to find naturally occurring xenografts and into cancer cell lines that we can further validate this testing with. And many, so far, the couple of antigens we've looked for, we can find RNA products, but we've yet to determine whether we can see the particular protein product and the splice product that we can then use for further evaluation. But I think we need to go around this wheel many more times and find more TCRs and evaluate them for this sort of function. Our hope is that one such TCR, recognizing one such target, might be useful not only in treating many patients with advanced prostate cancer, but might also cross over into advanced lung cancers, bladder cancer, and other epithelial cancer types in which the signaling milieu will make the cancer antigen more dominant and prominent and recognizable by these unique TCRs. So one of the things that's been rate limiting through this whole process is the finding these rare T cells bearing the TCR that's reacted to specific peptides that we're interested in. And so we're always looking for new ways to define and isolate specific T cells and their receptors that might turn into therapeutic entities. And this is just a nice list of a couple of the existing techniques, such as MHC tetramer, CD137, an activation marker, or the Clint-seq process that I talked about before. They have pros and cons, but each one is really limited to a limited spectrum of what's being measured. This one's affinity-based. This place, you can have several antigens at the same time. This one's based on cytokine production. But what seemed to us would be better is if you had a multi-parametric platform to isolate and define TCRs with greater specificity and sensitivity. And fortunately for us, Dino DiCarlo, who works here at UCLA, and I interact and talk and found out about a process that he had developed for making a very interesting micro-nano platform for evaluating cell interactions on solid surfaces. And it's basically called nanovials. He can manufacture these by a flow technique in which you end up with a tiny particle that's made up of PEG and gelatin. And then with specific cross-linking and removal, you end up with a particle that is PEG on the outside, gelatin on the inside, which can then be modified to contain active biotin sites, which serves as a target for adding just about anything else you want that you can molecularly produce to be coupled to this biotin site. You can get a lot of different molecules, for example, up to 10 to the 7th, 10 to the 8th MHC molecules for each one of these individual nanovials, and you can put multiple products along the surface of the nanovial, not just MHC, but other antibodies, other binding entities, and you really can let your imagination go wild for the types of things you can do. But what we've done with Dino's lab, and this is particularly the work of Felice Koo and my student Mao working in the setting, is to use antibodies which would capture the products secreted from an activated T cell, activated by the MHC T cell interaction, that would define the state of activity of the cell and the particular cytokines that are released, such as interferon gamma, IL-2. You can put various combinations together. And this technique has really worked out very nicely. Here's just a kind of an overall diagram of what would happen. So you've got a peptide MHC, interferon gamma, you find the right T cell and a rare cell among a population of cells that would bind. You could activate, secrete the cytokines. And most importantly, these particles are perfectly fine to be sorted and collected, and the cells through this process are viable, and you can take them off the beads of the nanovials and regrow them if you want, resort them, re-isolate them, do a lot of things with them. Just a couple of data points to show. Here's an example of the number of live cells loaded, percentage in nanovials. It's really very, very specific as you capture in this particular, a very strong TCR modified population of cells. It's 1G4 for a specific antigen peptide of NY-ESO, but it's very quantitative. You can also get very selective activation of the T cells here, showing gamma interferon positive cells among the population, looking at two double parameter sort here, the blue cells denoting very high levels of interferon gamma release. You can also show that you can find and bind cells at a level of about 1 in 25,000, 1 in 30,000, using this sort of spiked in control setting on this logarithmic scale. And I think really impressively, even if you use TCRs of varying affinity, and we had a series of TCRs we had previously isolated against NY-ESO peptides, these high to low affinity TCRs can be captured with this technique with almost 100% efficiency. So we're very much in favor of of switching our efforts to this particular technique, and we're doing so currently. So where we are with this is that we've shown that we can define the particular target peptides. We've shown that we can isolate TCRs, reactive against them. We've shown that we can modify and enhance the function of TCRs by using technology such as the catch bond. And we now have to go ahead and credential many of these TCRs for their function in vitro and in vivo model systems, and then consider some of them that might be appropriate to take on into the clinic. So I'll stop here and thank the many people who have worked on this. I try to mention the key people as we went through the talk, but I think the most important message I have is that this type of work absolutely requires collaboration with people who can bring expertise and to the type of analysis and discovery that we're most interested in. And so I'll stop here and be happy to take any questions. Okay, thank you.
Thank you so much. That was a fantastic presentation, Dr. Very. We do have a few questions. Let me open up the Q&A box over here. Ashish is wondering if you could maybe elaborate a little more on the PSCA that you described earlier and how it is being used as a target.
PSCA, well, that's a long time ago when we discovered PSA. It's an interesting story of how long things take from discovery to get into the clinic. PSCA was discovered in my group in the mid-1990s. It was part of the intellectual property that went out of my lab, along with other materials and discoveries, into a company called Agensis, which was formed here in Los Angeles with colleagues Ari Beldegrin, Rob Reiter, Charles Sawyers, and others. That company, took advantage of PSCA and made very good antibodies for it, and took it into credentialing. And eventually, the company was sold to a large pharma company, and then they didn't really prosecute it very well. And so now it's in the public domain, and there are several new smaller biotech companies that are working on PSCA as a target, and some academic labs, including my colleague Steve Foreman and Saul Priceman at the City of Hope, that are evaluating how it might work in advanced prostate cancer. They have some on-target effect, but they also have some side effects. And so I think they figured out ways to limit the side effects a little bit, and then are still considering it as an active target for cancer research. So 1995, 96 to now, it's the usual 20 years to see a basic science discovery make it into the clinic. But that's what's happening with PSCA as far as I know.
Great. Next question is from Stan Gu, a former postdoc in Miles Brown's lab and Dr. Shirley Liu's lab, now at MD Anderson. He's wondering, for the peptides derived from alternative splicing, do they recur in different patients, or do each patient have their own unique repertoire of alternatively spliced peptides?
I don't think each patient is unique, but we certainly wouldn't expect to see every patient with a histological definition of castration-resistant prostate cancer or neuroendocrine-like small cell cancer having the precise set of peptides. Nor would we expect that any of the particular spliced peptides would be restricted only to prostate cancer. In fact, one of the major things we've observed is that advanced small cell neuroendocrine featured prostate cancer is so similar to small cell lung cancer. It's really astonishing to me, just by gene expression data and by the splicing, how many examples we can find that would cross over between cell lines derived from small cell prostate cancer or cell lines derived from our Part B system, or cell lines derived from xenografts of advanced prostate cancer then grown as cell lines. And so I think what we're going to be looking for here are peptide targets that may be valuable in a range of cancers, but not necessarily every patient with that definition of cancer.
Thank you so much. Dr. Miles Brown would like to compliment you on your beautiful talk. And he's wondering if you have tried to discover TCRs for oncogenic mutations such as FOXA1.
We haven't done that. That's a perfectly fine idea. I don't know if the mutation of FOXA1 would be restricted by a specific allele. It was a little daunting to think about studying, other alleles of HLA. I mean, you double the amount of work you have to do. But with the new technology that I described at the end, in which you can multiplex and create these little nanovials with whichever HLA type you want, with whatever capture antibodies you want to detect different stages or surface antibodies on reactive to the T cell, I think expanding the range of targets is now definitely feasible. There's a lot of people working on activated mutations, for example, in KRAS. There's a number of publications on that and companies that are working on it. I think we'll probably concentrate on as the output from the general spliceosome as we get more and more refined data. But thank
you. A question from Kenichi Shimada. Using 17 TCRs targeting 8 peptides uniquely expressed in cancers, roughly what % of CRPC patients are expected to respond? And also, how long do you expect it'll take eventually when you establish the pipeline to identify new epitope, make it into a new therapy?
The time expectation for that is, boy, I hope not as long. As I'll give you an example. BCR-ABL was discovered or demonstrated in the early 1980s, okay, by my lab, work at the RNA level by others, and at the DNA level by other groups as well. So that constellation of findings clearly defined BCR-ABL as a potential target. Well, it took till 2000, 2001 before imatinib entered clinical progress, ended clinical trials and entered clinical use. So, roughly 18, 20 years. Almost the same timeline for Bruton's tyrosine kinase and ibrutinib, okay? Hopefully, we can bring things a little faster, but each one of these credentialing and getting things into clinical trials just takes some time and obviously a lot of money. So what we're trying to do is generate enough candidates that, as they drop out for one or another reason, we'll be left with a few, okay, that are worthy of trying to take into the clinic. And how long that will take, I don't know, but I hope it's not 20 years.
All right. The next question is related to your PARC-CB model. So you have described other models with different drivers and MYC, and so are you at some point also planning to do the same time course studies for these different drivers of neuroendocrine prostate cancer in combination?
I'm not sure we'll do other drivers, and try to do a cross evaluation. One of the nice things is having good postdocs that develop things like this, like Jun-Wook Park, who's now at Duke University as an assistant professor. And those are some of the questions that I think he'll be more interested in answering. So I think we'll probably leave that to him and others to do, but I think concentrate on the most potent model and look at this kinetics and try to look at gene control as to who's regulating the process. We can correlate what happens, who's expressed, but we don't know yet in this wholesale genomic rearrangement who's really organizing and driving that. We know we can initiate it, and then we can watch it proceed. But we don't yet know for the various stages of conversion from the adeno through to the small cell neuroendocrine, which specific transcription factors are obviously required to make it happen.
Those are all the questions in the Q&A. That's great. So I would, on behalf of the Connect Science community, I would like to thank you for taking the time to present. And I look forward to more exciting research coming out of your lab, and good luck with your future research endeavors.
All right, thanks very much. And I'll talk to you and the other postdocs, I guess, later this afternoon. All right, take care.
Thank you.
Bye-bye.
I'd also like to take a minute to thank all of the attendees for being here today. Next week, same time, we will be hosting Dr. Howard Sealer from Hebrew University, who will be speaking on the fundamentals of DNA methylation in development and environmental adaptations. See you soon. Thanks.